class, we in fact had a lot of question and answer session, and it was with very informal, where students kind of took their time to do different questions and different things and try out different combinations, and then they um, had it checked with me. So basically, we spent time in class doing things, okay, finding possibilities and combinations, and then arriving to some conclusions. So that is what we did in class. And I encourage you all to go ahead, have a pen and paper in your hand as well, try out different combinations, because you see, we don't have enough time really. So you only have 9 days until your final exam. This is part of the final exam. So if there are any confusions, I would suggest, recommend, and urge you all to please go ahead and try this out. And if there are any questions, confusions, just ask me now after the end of the class. Or if it's really very confusing, just stop me there and then and ask me right away. Because this is what we did in physical class as well. There were confusions to a lot of students. I mean, the topic is fairly simple. We're talking about blood transfusions. Everybody understands blood groups and everything, but in some places it gets rather tricky. So I would recommend all of you to please do go ahead, have a pen and paper on your hand with yourself, and try different things out when we talk about them. Okay, so let me share my screen and we will proceed from there on. Okay, here we are. Blood transfusion. We are keen on understanding. There it is. Okay. So we are keen on understanding different components of blood, because different components of blood can be transfused. Of course, blood itself as a whole can be transfused as well, but sometimes we are also interested in uh, transfusing only certain components of the blood. For example, platelets. You are all familiar with the platelets. As a nation, we've gone through this, and especially in dengue, where you've seen how platelets begin to fall, and then there's an emergency of getting platelets up and ready for the patient. So there are different components in blood basically, and that could be transfused separately, and the blood itself as a whole as well. And those different components are red blood cells, white blood cells, plasma, platelets. Each of them have a function of their own. Red blood cells, majorly, they contain hemoglobin, and the core purpose of that is to transport oxygen. So they are basically oxygen carriers. We can think of red blood cells as oxygen carriers. And um, the idea is to, basically ensure that oxygen levels are distributed throughout the body, and the primary source of that is blood. So red blood cells are in abundance in your blood, and they're the oxygen carriers. Next up, we have plasma. So essentially, the thing that makes your blood as a liquid medium, something that is basically carrying the flow of in everything, that is plasma. It's basically the liquid that we say is carrying all of the cells and everything within the blood. So that itself is plasma. Then we have these white blood cells, and we categorize these white blood cells into phagocytes and lymphocytes. So there are two kinds of white blood cells that we talk about usually. The phagocytes kind of engulf the material, which is put together by lymphocytes. So lymphocytes, they tend to bind together the any foreign particle, any foreign intruder in your body, if you will, and those clumps that have been lumped together by those lymphocytes are engulfed, eaten up by the phagocytes. So those two kind of white blood cells, they function together, and they serve as a immune system in your body, as a policeman of different intruders, if you will, in your body. So that's a kind of an analogy that you can go with: policemen and intruders in your body as far as the immune system is concerned. And then you have these platelets. The platelets, they help to kind of clot the blood in the body, and these are very instrumental in stopping blood flow. Without these, you may have your blood flow going on about from any wound indefinitely, and that often could lead to uh, say loss of a significant loss of blood, which could eventually prove very fatal and dangerous. especially when you have certain injuries or wounds or accidents. Okay, so that being said, this was the base setup, that premise that we had in today's session as well, that we understood blood components, we understood what they are, what their function was, where are they produced. Platelets, we know, are produced in bone marrow. As a matter of fact, I am supposed to share 3 links to 3 videos basically on YouTube, which are very nice, very instrumental in your learning, and they will enhance the concepts that we are going to discuss today. So at the end of the session, I will share those 3 links with you, and you are encouraged to go over them. They're going to build on that. So our discussion is going to be built upon on that. What we are here to discuss next up is this man. I keep on forgetting his name. Dr. Carl Landsteiner. Uh, it was in 1900 that he figured out that some people, after getting a transfusion, tend to live while others tend to die. So he observed that some people lived and some people died after transfusions, and that was rather intriguing, rather shocking, and rather interesting, if you will. That why does it happen that some people after receiving blood end up dying, okay, whilst others tend to live? So and then his research is surrounded with this around this observation, and 30 years later, in 1930, he received his Nobel Prize exactly for this. Exactly for what? Exactly for identifying and understanding that there are certain blood groups in humans, and as a matter of fact, they are about 33. I'm not sure if you're able to see the end of the list. I will try to move this around, and yeah, I hope you're able to see the list. All
Yes,
the
sir.
Yeah, okay, thank you. So there are about 33 blood groups, right? Blood group systems for humans. Okay, what we are interested in are the ABO system, the number one, and the RH system, the Rhesus. Okay, so these are very common and often typically used, these two systems. The rest of them do exist, but our discussion is going to be limited to these two. Okay. Then we move on to uh, certain understanding, certain concepts related to the nomenclature, related to terminologies. We just want to get them out of the way, understand what they are. So basically, I want everyone to be on the same page that there are certain things called alleles. Okay. Alleles are simple singular elements, singular proteins, if you will. They are just one. Okay, so they are just simple one. We're not talking about a group, we're just talking about one contribution from one parent, one kind of a gene. So that is an allele. Okay, a combination of two of them, that is a genotype. The combination could be of both of the same nature, both capital D, um, one capital D, one small d, one small d, one capital D, both small d's. Okay, so they could be dominant or recessive, but irrespective of that, two alleles, they make up a genotype. So in fact, if I put it simply, these are simply the possible combinations. Okay, so there are 4 possible combinations. There are 4 kind of genotypes that could be made up from 2 alleles that we have up on top. Okay, when these genotypes, once you have these genotypes ready, the next stage or the next step in this pipeline is that you determine what exactly is going to express, as a gene, what exactly is going to express. So here we have 2 dominant alleles, and we know in this genotype that either one could express itself. So it will be a capital, double D. So it is homozygous because it is same. The small d's are also homozygous because they're same. And in both of these extreme cases, we know that the expressive result would be the same, the capital double D's and capital small d's. However, in the central 2 examples that we have, um, we say that the dominant allele, which is the capital D, is going to express itself. All right. So basically what we say is that the phenotype is going to be a capital D in this case, a capital D in this case, and a small d in this case. So when you talk about the expression of the phenotype, we have a dominant expression here, we have a dominant expression here. There's nothing dominant here, so we say a recessive um, gene has been expressed. So this entire example in itself is for a sickle cell anemia, okay, a cystic fibrosis, that these genes, they remain unaffected. However, the recessive ones, they are the ones that are affected. But we're not into the example right now. What the goal for this slide is for you to understand that alleles are individual elements here, genotypes are their possible combinations, and phenotypes is the expression of these possible genotypes. Okay, so here we do not know what will be expressed as a gene, but when we talk about phenotypes, we are exactly 100% sure that this is what has expressed, this is what is the result of a possible genotype. Okay, so I hope that
Excuse me sir,
G, please.
सर ये जो रिसेसिव phenotypes हैं ये these phenotypes are always affected by disease
G, bilkul, bilkul, that is the point
and the
of it.
dominant ones are not affected.
They are not affected, लेकिन जिन लोगों
Sir,
को
but
ये होते हैं,
why?
they are the ones that are affected. Maybe they are not strong enough, maybe they are more immune, maybe they are not able to fight back, maybe it's an hereditary thing. At this point in
Okay.
time, it does not concern us because we're not into understanding sickle cell anemia right now. We care about understanding what are alleles, what are genotypes, and what
Okay.
happens to the expression of these genotypes. They are called phenotypes. So we want to
Sir,
know.
genotype and genes both are same.
You could argue they are both same, yes.
Okay.
Okay,
Thank you.
but that is it for this slide. And then we went on to a small class exercise where we said, and if I am allowed to share my screen, let me switch it to the one moment. Okay, right. So then we move towards a simple example. We built our discussion through this, and we said that assume that we have this example, and if you know your blood group, that would be great. We said that we are going to, let me write this down and zoom this up. Okay, so we said we are going to go ahead with a we have said we are going to go ahead with an example. We've been set this up as father and mother, on father on this side, mother on this side, and assume if you know your father's blood group, you go ahead and write his blood group is A. If you know your mother's blood group, you say it is B. And then we looked up into different combinations and we figured out that AB is a combination, AB is a combination, AB is another combination, AB is another combination. So we figured that in this way we could find out that if a father has a blood group A and a mother has blood group B, like this, it would result in possible blood group combination of their children like these, and they're all same in this case. But we said that blood group A could be expressed as this, and blood group A could also be expressed as AO. What this essentially means is that your grandparent, so if your father has AA, which means one of them is coming from his father and the other from his mother. So if you have AO, it's possible that your father's parents have different contributions. Instead of AA, one of them might have an AO combination. Okay, so we then looked into that and we figured out that if they have a different combination, it kind of changes the results somewhat. Okay, how does it change the result? Because now you see the combinations are slightly different. How are they different? They are different because of this O, we have a BO and a BO expressed here. And we developed this concept basically that B, blood group B, can be expressed in two ways, as a BB and a BO. with B being the dominant in case these two exist together. Okay, so if you have a B blood group, you may not necessarily have a BB, it could also be a BO blood group. Okay, uh, with B being the dominant one. So we say that in terms of hierarchy, B and A are always more dominant than O. All right. At this point, I gave everyone enough time. You can take 5 minutes to draw this for yourself. If you know
Excuse
your
me, sir.
really,
Sir, some people have O negative blood group. It means that they're having O dominant.
right now, we will not talk about positive and negatives. That is yet to come in about 10 minutes time. But
Okay.
as far as O is concerned, I would recommend you to give this a try. How? Uh, in fact, let's do this as a class exercise. Everyone can go ahead and try. Rather, if you do for your, just do two exercises, okay? One for your own blood group and one for this case for O. Okay. So identify a
Okay.
case where you will have a blood group O. Okay. How can you get a blood group O? If you have a if you have AO, you will not have a blood group O. If you have BO, you will not have a blood group O. In both
Not.
of these cases, you will have A or B. Okay. So the only way to get a blood group O is to have OO. All right.
Yes.
So
Yes.
how can you get that? What are the possible combinations? So you must have a contribution of O from your father and a contribution of O from your mother. So you must have both of these contributions from both of your parents to get OO, and as a result, you will have blood group O. Okay, so that we know. The rest could be different. Okay, it could be A, it could be BO, it could be BO here, it could be anything. Okay, so the other combinations could be different combinations, but here is the important thing that you must have one contribution from father and mother in order to get um, this blood group O here. Okay, so that is a key observation that we also had in class today. I urge you all to try this for yourself. And if you don't know your parents' contribution, but you know your own blood group, you could trace back your parents' blood group type from their mother and father in this way. Okay. So if there's anyone who wants to do it, you're welcome to do it here. Or if you want to do it on your own notebooks, you can do it on your own. No problem with that. Um, any questions about this so far? Because we're going to go ahead and build up on this. Okay, I take it there are no questions. Next up, we talked about some basic things, that there are these proteins called antigens. We Yeah, this is where we talked about antigens. Okay, so we said there are these proteins that are present on the surface of of red blood cells. Okay, so when you talk about blood group A here, we say it has a certain antigen, certain protein binded on the surface of it. Okay, and these proteins, we call them antigens. Blood group V would have a different set of antigens. I can do this again. Yes. Blood group AB, the third blood group, would have both of these antigens, and blood group O would not have any antigens at all. Okay, so these antigens are basically part of your red blood cell, they're on the surface of it, and these are basically differentiating between different blood group types as well. So when someone has a blood group A, we say they have these antigens, and they have certain antibodies as well in their immune system. The antibodies that this blood group has are anti-B. Okay, antibodies, anti-B antibodies, these are the antibodies that they have. What does this mean? This means that in case blood group A sees any antigen which is not present in its own blood group, such as these blue ones, then it's going to treat them as an intruder, as a foreigner, and it's not going to accept it. Okay, so it has anti-B antigens. So if someone donates blood group B to a person with blood group A, because of these antibodies which are anti-B, this person is not going to accept this blood, and as a result there's going to be an immune reaction. And that immune reaction, there are many names for it, but one of them is agglutination. Okay, so agglutination is basically the clotting or the clumping together of different of blood cells together, and that happens because of the massive mismatch between the two kind of bloods. Okay, so okay, Harun, I'll go over this again. So we say that a blood group has antigen on top of the red blood cells, and there are some antibodies that are present in the because of the immune system in the bloodstream, and these antigens are specific to each blood group. So blood group A has these antigens, blood group B has these antigens, blood group AB has these antigens. They have both of them in A and B. Blood group O does not have any antigen. Okay, so when blood group A receives blood from a person with blood group B, it should be problematic, and it is problematic. Why? Because blood group A has these antibodies as well. Okay, so the antibodies present here are anti-B antibodies. So it cannot receive blood group from B because these antibodies are going to attack these antigens present on blood group B, red blood cells. Okay, so these antibodies are going to attack the antigens here. They are going to attack these cells here because they're against them. Okay, similarly, blood group B has anti-A antibodies. So if it gets exposed to blood group A, these antibodies in this person's blood are going to attack these cells. Okay, and as a result, might lead to agglutination, which is clumping and clotting of blood and thickening of blood
Sir,
vessels. G, G, G.
sir, a person having a blood group B can donate its blood to the person having blood group AB.
Just hold on for a minute and we will answer this exactly. Just 1 minute. Let me complete this just for
Okay.
completeness sake. Then I'll answer this. The next one is the blood group AB. It has both of these antigens on its surface, but it does not have any antibody. Okay. So if it does not have any antibody, it means it can receive blood. It can receive blood from A, it can receive blood from B. It does not have any antibodies which are going to attack them. It does not have any antibody which is going to attack A. Okay, so you just ask this question. Yes, B can donate to AB. Why? Because it does not have any antibody to attack this antigen on B. Okay. When you talk about blood group O, it does not have any antigen on its surface, but it has these antibodies A and B. So these antibodies are going to attack from antigens on B and antigens on A if they donate to O. Okay. Similarly, if AB donates to O, these antibodies are going to attack them. Okay, because it has antibodies for both of them. Um,
सर ओ वाला जो ब्लड ग्रुप है उसके पास ए और बी दोनों के एंटीजन्स हैं और वो इन दोनों से ब्लड ले सकता है.
नहीं, इसके
सर
पास
ये मुझे
antigens तो
क्लियर
है
नहीं
ही,
हुआ ओ
इसके
वाला.
पास एंटीजेंस है ही नहीं. यहाँ देखें कुछ भी नहीं बना हुआ. इसके पास
ओके
एंटीजेंस है ही नहीं. एंटीबॉडीज इसके पास दोनों
ओके
है. ओके, सो एंटीबॉडीज
ओके
दोनों.
ओके ओके
एंटीबॉडीज दोनों होने का मतलब ये है कि ये इसके पास अगर ए की एंटीजन आएंगी तो उनको भी अटैक करेगा, बी की एंटीजन आएंगी उनको भी अटैक करेगा, क्योंकि इसके पास दोनों की
ओके
एंटीबॉडी. ओके, सो इट
ओके
कैनॉट रिसीव फ्रॉम ए बी. और अगर ए बी डोनेट कर दे तो ए बी तो दोनों ही एंटीजन यहाँ लगे हुए हैं और इसके पास तो एंटीबॉडीज भी दोनों की हैं, तो ये दोनों ही उस पर अटैक कर देंगे, तो ए बी तो कर ही नहीं, बिल्कुल नहीं कर सकते हम. ठीक है,
Okay, okay,
तो
okay,
इसलिए
okay.
ओ को कोई रिसीव नहीं होगा. नाउ एज एन एक्सरसाइज, you are all encouraged to, okay, I have done the first one here. The rest of them you can take 5 minutes to complete them, and then I'll show you guys. This is the phenotype. Remember, phenotype क्या है? Which has been expressed. ये हमें पता चल गया, एक्सप्रेस हो गया है, ए ब्लड ग्रुप है. It could be AA, it could be AO. Irrespective of that, we know the phenotype is A. ये यहाँ ब्लड ग्रुप A ही है. अब A हो, AO या AA हो, those are दो two genotypes. Okay, वो हम बात नहीं करें. I've given you the phenotype कि we know this is blood group A. Okay, जो भी डोमिनेंट था A O में से A था तो A हो गया, AA था तो A, एक ही डोमिनेंट, दोनों ही डोमिनेंट है तो ब्लड ग्रुप A ही बनता है. तो वी नो द phenotype, दिस इज ब्लड ग्रुप A. अब एंटीबॉडीज की बात हो रही है तो ए के अंदर हमें पता है दे हैव एंटीबॉडीज बी इन द सिस्टम. ओके, इन द इम्यून सिस्टम, ब्लड स्ट्रीम में वो एंटीबॉडीज बी होती हैं, एंटी बी एंटीबॉडीज उसके अंदर हैं. यहाँ पर डोनर्स हैं ए बी ओ, एंड यहाँ पर रेसिपिएंट्स हैं, तो ये रेसिपिएंट है. तो वी आर टॉकिंग अबाउट वेदर, we are talking about whether blood group A can receive, it's a recipient ना, so whether this person can receive blood from A. बिल्कुल, ब्लड ग्रुप ए है, वो ए ने ए को ही कंट्रीब्यूट करना है, इट कैन एब्सोल्युटली रिसीव, कोई मसला नहीं है. लेकिन वेदर इट कैन रिसीव ब्लड फ्रॉम बी, अगर बी डोनेट कर दे इसको ए को, तो दैट इज एन इशू. क्यों? क्योंकि ए के अंदर एंटी बी एंटीबॉडीज पड़ी हुई हैं, वो इसकी एंटीजेंस को अटैक कर देंगी, तो यू कैन नॉट रिसीव इट. इसी तरह ए के अंदर एंटी बी एंटीबॉडीज पड़ी हुई हैं ए के अंदर, वो जब बी के एंटीजन आएंगे तो उनको अटैक कर देगा. ए बी के अंदर रिमेंबर दोनों एंटीजन हैं, तो बी के अंदर जो एंटीजन है उनको अटैक कर देगा, इसलिए यू कैन नॉट रिसीव ब्लड फ्रॉम ए बी. लेकिन ओ के अंदर कोई भी एंटीजन नहीं है, तो इनकी एंटी बी एंटीबॉडीज जो ब्लड ग्रुप ए के अंदर है, इट डजेंट मैटर. वो ये एंटी बी एंटीबॉडीज, इसको है ही नहीं कोई एंटीजन जिसको वो अटैक करे, तो इट कैन इजिली रिसीव ब्लड फ्रॉम ब्लड ग्रुप ओ. ओके, तो यहाँ पर वी हैव फिगर्ड आउट कि ब्लड ग्रुप ए के अंदर हमें पता है एंटी बी एंटीबॉडीज हैं, और वो किन किन से रिसीव कर सकता है ब्लड. नाउ 5 more minutes for everyone to complete this table for blood group B recipient, for blood group AB recipient, and for blood group O recipient. You can just take a few minutes to complete this. Any volunteer who's done the second one can open their mic or and maybe discuss this for recipient B. जी, no volunteer.
यस सर.
जी बताएं.
सर जो बी है ब्लड ग्रुप, वो ए वाले से नहीं ले सकता क्योंकि उसमें एंटीजन होंगी.
Okay, great.
सर इसी तरह ए बी वाला भी नहीं ले सकता, बी वाला और ओ वाला बी से ले सकता है.
नहीं, आप पहले बी के लिए बताएं. बी ए से नहीं ले सकता, बी बी से ले सकता है, बी ए
बी
बी से ले सकते हैं या नहीं?
से ले सकते हैं.
यस
बी
और नो?
ए बी से नहीं ले सकता, नो.
क्योंकि सर उसके अंदर बी एंटीजन भी हैं, एंटीबॉडीज भी हैं, तो वो नहीं ले सकता ए बी से.
यस सर, ए बी से नहीं लेगा.
लेकिन रीजनिंग विषय आप सही करें, बिकॉज उसके अंदर ए एंटीजन, बी नहीं ए एंटीजन है, इसके पास एंटी ए एंटीबॉडीज हैं बी के पास,
ओह यस सर.
तो वो ए एंटीजन
यस.
को अटैक करेंगी. ओके, तो ए एंटीजन को अटैक करेंगी जो कि ए बी ब्लड ग्रुप के अंदर है, इसलिए वो नहीं ले सकते. एंड हारून फाइनली क्या है,
बी ओ से भी ले सकता है.
क्योंकि कोई वो एंटीजन है ही नहीं जिसको वो अटैक करे, तो वो उससे भी ले सकते हैं. ओके, ठीक है, so that makes sense. Anyone with the AB, anyone other than Harun? Harun has had his turn. Anyone else wants to volunteer for AB? Let's do this quickly. Anyone?
सर ए बी में एंटीबॉडीज कोई भी नहीं होती, जिसकी वजह से वो सबको डोनेट,
सबको डोनेट करके सबसे रिसीव कर लेगा.
सबसे ले सकता है सर.
बिल्कुल, AB में कोई एंटीबॉडीज नहीं है, किसी को उसने अटैक करना ही नहीं है. यहाँ पे कोई एंटीबॉडीज AB की नहीं है, उसने किसी को अटैक करना ही
यस सर.
नहीं है. वो A से भी रिसीव करेगा, B से भी करेगा, AB से तो कर ही रहा है, और O से भी रिसीव कर लेगा. ओके जी. Next up we have O. Anyone, any volunteer for O? जिसका blood group O है वही बता दे. Anyone?
सर ग्रुप ओ के अंदर हमारे पास बोथ
आपका blood group
एंटीबॉडीज
O
हों.
है?
नो सर.
You had your turn. Let someone else participate. Anyone else? No one else. Okay, मैं बता दें, आप ही बता दें.
सर ब्लड ग्रुप ओ के अंदर हमारे पास एंटीबॉडीज बोथ हैं, ए बी और बी बी, तो वो रिसीव नहीं कर सकता ए से
ए से भी नहीं, बी से भी नहीं और ए
और
बी से भी.
ए बी से भी नहीं, मगर वो ओ से ही बस रिसीव
हो
कर
सही कर
सकता.
सकता है. ठीक है, तो वेरी नाइसली पुट. ये इसी तरह ही है. यहाँ पर some people were getting confused in the physical session, and the reason for that was they were looking at the donors from the donor's perspective. आप लोग confused नहीं हो रहे, there are no questions, well and good, but I will reiterate here that when you are trying to make this table, please make this table based on the recipient's perspective. ये जो एंटीबॉडीज हैं ये रेसिपिएंट के सिस्टम में है, उसकी बॉडी के अंदर है. जो ब्लड ग्रुप आ रहा है वो अपने साथ एंटी ए नहीं लेके आ रहा है. ये डोनर अपने साथ एंटीजेंस वाले सेल्स तो भेज रहे हैं, एंटीबॉडीज नहीं भेज रहा. तो डोंट गेट कंफ्यूज. सम पीपल आर गेटिंग कंफ्यूज कि ये एंटीजन इसके साथ एंटी ए भी तो है, जब ये इसमें जाएगा ब्लड तो ये एंटी ए इसको मार देगा इन एंटीजेंस को. नो नो, वी आर नॉट टॉकिंग अबाउट दैट. दीज एंटीबॉडीज आर नॉट कमिंग इन हियर. The donor is only providing the blood with the antigens. Yes, cells provide कर रहे हैं with the antigens. आपने रेसिपिएंट के पर्सपेक्टिव से एंटीबॉडीज को देखना है. रेसिपिएंट देखना है, उसके देखना है उसके सिस्टम में क्या एंटीबॉडीज हैं, उस हिसाब से डिसाइड करना है व्हाट इट कैन एक्सेप्ट एंड व्हाट इट विल रिजेक्ट. ठीक है, तो दैट वाज जस्ट अ क्लैरिफिकेशन. विद दैट इट शुड नॉट बी एन इशू. अच्छा, नेक्स्ट अप वी said that when foreign antigens are discovered, antibodies are made by B cells of immune system. Immune system में we say there are B and T cells. They are both
एक्सक्यूज मी सर.
lymphocytes. जी जी.
सर वो मेरा इंटरनेट प्रॉब्लम हो गया था, तो जो आप ओ वाला एक्सप्लेन कर रहे थे वो मैं नहीं सुन सकी. मतलब सर उसको रिपीट कर देंगे काइंडली.
Don't worry,
कि
मैं बताता हूँ. Hold on.
ओके, थैंक
We
यू.
were saying that blood group O has both of these antibodies, anti-A and anti-B, which means that it will attack antigens A and antigens B. जिसका ये मतलब है कि ए से तो रिसीव नहीं कर सकता, ना बी से रिसीव कर सकता है, ना ही ब्लड ग्रुप ए बी से रिसीव करता है, क्योंकि इनमें एंटीजेंस लगी हुई हैं. सो इट्स एंटीबॉडीज आर गोइंग टू अटैक दीज एंटीजेंस. एज अ रिजल्ट इट कैन नॉट एक्सेप्ट फ्रॉम दीज थ्री. इट विल ओनली एक्सेप्ट फ्रॉम इटसेल्फ. ओ सिर्फ ओ से एक्सेप्ट कर सकते हैं. इसके बाद मैं ये बात कर रहा था कि आप हमेशा रेसिपिएंट के पर्सपेक्टिव से बात किया करते हैं जब भी ये देखना होता है कि कौन डोनेटर रिसीव कर सकते हैं. रेसिपिएंट के सिस्टम के अंदर एंटीबॉडीज हैं. डोनर एंटीबॉडीज नहीं अपने साथ डोनेट करता. डोनर अब सिर्फ सेल्स दे रहा है, उसके ऊपर एंटीजेंस लगी हुई आ रही हैं. ओके, डोनर अपने साथ ये एंटीबॉडीज भी भेज दे तो क्या होगा, वो एंटीबॉडीज अटैक करने शुरू कर देंगे होस्ट के सेल्स को. तो ये एंटीबॉडीज नहीं आती, उसके सेल आ रहे हैं. जब हम बात कर रहे होते हैं तो हमेशा रेसिपिएंट के पर्सपेक्टिव से बात करते हैं. रेसिपिएंट का सिस्टम है, रेसिपिएंट इज द होस्ट. रेसिपिएंट के अंदर इम्यून सिस्टम है जिसने ये एंटीबॉडीज प्रोड्यूस करनी है, उन्होंने इंट्रैक्ट करना है, उन्होंने कैप्चर करना है और ईट अप कर जाना है, या जो भी मैकेनिज्म होगा बॉडी का टू एलिमिनेट द इंट्रूडर्स या फॉरेन एंटीजेंस. तो वो रेसिपिएंट का हवाले से आपने बात करनी है. रेसिपिएंट की एंटीबॉडीज देखनी है और फिर डिसाइड करना है अच्छा ये एंटीबॉडीज किसको अटैक करेंगी, किसको एक्सेप्ट करेंगी, और उस हिसाब से आपने ये ड्रॉ करना है. तो इसको रटने की जरूरत नहीं है. फाइंड द लॉजिक इन दिस. लॉजिक यही है, उसकी एंटीबॉडीज देखें रेसिपिएंट की और देख लें किसको वो एक्सेप्ट करेगा या रिजेक्ट करेगा, एंड बेस्ड ऑन दैट यू कैन मेक द स्टेप. ये बात कर रहे थे हम. उसके बाद हमने कहा कि when foreign antigens are discovered, antibodies are made by B cells of immune system. This is a fairly simple thing to understand. B and T cells दो तरह के cells होते हैं immune system में. They're both of lymphocytic nature. ठीक है ना, हमने कहा था शुरू में lymphocytes and phagocytes दो तरह के white blood cells हैं immune system के. वही basically they're the heart of it. B cells are the kind of cells that make these antibodies. Okay. But the core
सर
idea in this statement, जी जी,
सर ये एग्लूटिनेशन क्या है?
agglutination थी clumping of सेल्स उनका
ओके,
क्लम्प हो
तो
जाना।
ब्लड ग्रुप
क्यों होते हैं क्लम्पिंग ऑफ सेल्स? वो क्यों इकट्ठे जम जाते हैं, इकट्ठे होना शुरू हो जाते हैं? इम्यून रिएक्शन के नतीजे में, जब ब्लड ग्रुप्स मैच नहीं कर रहे होते हैं।
ओके
आपके वो बेसिकली your immune response, there's an immune response. These antibodies tend to eat up those cells, those foreign cells. Those foreign cells cannot mix up with your cells, with your host cells. And imagine a blood group B has been transfused into a person with blood group A, and leads to a reaction. That reaction, that is agglutination itself. Okay, where these cells tend to clump together and they become thick. It leads to blood becoming thick, coagulates your arteries and veins, and ultimately leads to cardiac arrest or death. Okay, so that was one thing. Next up, we said that for some reason this is not updating, but it's okay. I will go ahead. Next up, we went into the discussion for rhesus proteins. ठीक है, what we said was कि ये जो rhesus proteins हैं, ये भी एक प्रोटीन है. ओके, दीज आर ऑल्सो प्रोटीन्स. ये भी आपके इन्हीं सेल्स के ऊपर लगे हुए हैं. ये एक अलग प्रोटीन है. एंटीजेंस ये अलग एंटीजेंस है, ये अलग प्रोटीन है, rhesus protein. If the rhesus protein is present, we say that your blood group is positive. If the rhesus protein is not there, we say your blood group is negative. ठीक है जी. तो फेयरली सिंपल, तो बहुत आसान हो गया. बल्कि प्रेजेंट है तो पॉजिटिव है, नहीं प्रेजेंट नेगेटिव है. आपका ब्लड ग्रुप बी पॉजिटिव है, इसका मतलब है आपके ब्लड ग्रुप के सेल्स के ऊपर ये वाली एंटीजेंस तो हैं ही, बी की जो होती हैं, और इसके साथ और प्रोटीन्स भी बाइंड हुई हुई हैं, रीसस प्रोटीन्स. जिसके पास ये बाइंड नहीं हुई हुई वो बी नेगेटिव है. तो दैट इज द बेसिक आइडिया ऑफ दिस. जीनोटाइप्स फिर हमने लिस्ट की. जीनोटाइप्स, रिमेंबर व्हाट वाज जीनोटाइप्स? जीनोटाइप्स वर द पॉसिबल कॉम्बिनेशंस. दोनों डोमिनेंट है, दोनों रिसेसिव हैं, या दोनों का मिक्सचर है. ओके, फिनोटाइप क्या था? रिमेंबर फिनोटाइप वी सेड वाज अ फाइनल डिसीजन कि आखिर एक्सप्रेस होगा क्या. सो इन केस ऑफ बोथ वर डोमिनेंट, वी सेड दिस इज गोइंग टू एक्सप्रेस, पॉजिटिव है, आर पॉजिटिव है. एंड देन वी सेड अगर दोनों ही रिसेसिव हैं तो क्या होगा, आर नेगेटिव हो जाएगा. ठीक है, दैट इज गोइंग टू बी expressed. And then we said that in case they are heterozygous, the terms basically are homozygous if they are same, homo. Hetero is if they are different. Okay, so ये ऊपर first and third are homozygous. The central one is different. Okay, different alleles. So we different alleles in this genotype. Okay, remember the convention, the nomenclature, or the academic jargon, if you will. So these two alleles are different in this genotype, which makes this genotype as a heterogeneous genotype. The phenotype turns out to be positive. Why? Because we said that the presence of protein, this allele is always dominant as compared to the recessive one. So अगर इस तरह का combination, if you get a combination like this, you'll always have a positive or R positive rhesus protein expression. Okay, so that is what we say is there. Next up, okay, so remember this is about the protein being present. If you draw a comparison with the ABO system, presence of antigens on the surface. We have talked about presence of protein on the surface. Now we talk about the antibodies. यहाँ पर in ABO system, you know what antibodies they are. A में anti B, B में anti A, AB में there are no antibodies, O में there are both of the antibodies. What about the rhesus system? In the rhesus system, the positive phenotype does not have any antibody at all. There are no antibodies. But for the negative one, there is an antibody here, anti-positive, or there couldn't be anything else because there's only anti-R positive antibody. And if you think about this carefully, take a minute to think about this, and in fact, go back to, if I if I zoom out a little bit, yeah, go back to this sentence that we had on this side. When foreign antigens are discovered, antibodies are made by B cells of immune system. This actually makes sense. Why? Because imagine yourself, if you are R negative, you have a B negative or A negative blood group, you don't have a rhesus protein in your system. Okay, Your system does not recognize a rhesus protein. It does not know what a rhesus protein is. Okay, so it has never seen a rhesus protein. The moment it sees a rhesus protein, it's going to generate antibodies. Why? Because if you get transfused blood from someone with a positive blood group, you're A negative, you get blood transfused from A positive person, that is going to be the first time your body is going to see a rhesus protein in his blood, in his cell, in his blood cell. Okay, so and it's going to it won't know what it is. So as a result, it's going to generate antibodies against it, and it's going to treat it as a foreign antigen. It's going to fight against it, and that is why we say that R negative have anti R positive proteins. Okay, I hope that is clear. In the R positive case, you already have the protein. Okay, you would not need any antibodies. You get transfusion from a person with positive, you already have the same kind of proteins in your system. There's no issue in your system to identify the received blood as a foreign antibody or foreign antigen. So you wouldn't need an antibody. There wouldn't be any reaction into that. And if you receive blood from a negative person, from a negative person, okay, fine, they don't have a rhesus protein on their cell, so that's not an issue. You have the rhesus protein yourself. The absence of it is not an issue. You don't need an antibody for something that is not even there. So that is why we say they don't have antibodies, and here they do have anti R positive proteins. So this was our discussion that we had today, and at the end of this discussion we had one case study basically, and that was the we had a case study on the rhesus disease. So if there are any questions so far, please let me know. Otherwise we will proceed to the case study, a disease, a rhesus disease, and then we will wrap up our discussion. Any questions here so far? Okay, I take it there are no questions. So let's go to the rhesus disease problem. So what is the rhesus disease problem? We begin with an imagination here that we have a mother with R negative, rhesus is rhesus negative, whatever the blood group is, and a father who is rhesus positive. Okay, he has the protein, mother does not have the protein. When they conceive a child, okay, when they conceive a child, I've represented M for mother, F for father, and C for child here, M for mother here. So when they conceive a child, their child, what is the possible genotype when they conceive a child with mother being R negative and father being R positive? This is the possible genotype that you will have. Okay, one of them with the protein, the other without the protein, right? R positive, R negative. So when they have this genotype, it results in a phenotype that is R positive. Okay, so that's what we say, that when these two conceive a child, the child is going to have a R positive rhesus protein in the blood group. Okay, it will be rhesus positive. Now, if you think critically, you should come to a conclusion that this is a problem. I will let you take 30 seconds and think about this. Look at the antibodies that we have, and remember, this is a disease. This is a disease. See how the mother has R negative protein. In fact, that's the absence of protein. So mother is R negative, but the child is R positive. And when you think about it, the mother being R negative has anti R positive proteins. Okay, so what does that tell you? That during pregnancy, the mother's immune system should treat the child's red blood cells as a foreign antigen, because the mother has never seen an R positive protein and now she sees it. That should trigger an immune response, and that should lead to the destruction of these cells. So I want you to take 10-15 seconds to appreciate this, that this is a problem in fact. And that was the problem that we discussed, and we were trying to find a solution to this. So I hope everyone is clear on the problem. If there's any issue in recognition of this problem, please ask now. There is a problem. There's a basic problem in this. That the mother is R negative, the child is R positive. The mother treats the child as a foreign, foreign antigen, foreign particle, and during pregnancy, and that is an issue. Do you all agree? By the way, this is an issue.
Yes, sir.
Okay. So somebody does agree, but I think people who agree to this are in minority. There's only one person who agrees, the others don't. But you can take my word. This is a problem. Okay, so we'll move on. Uh, the the issue in this is then.
सर ये ऐसे गलत बात है सबके रूल फॉलो होने चाहिए सिर्फ एक बंदे के लिए नहीं होने चाहिए
Okay. I totally understand this is a problem. Don't worry. So uh, then the resolution to this problem is that what actually happens is that uh, that we need to recognize and go back to what we had a comment here, that whenever we were talking about these blood groups mixing and getting transfused, we were always supposed to consider the perspective of the recipient, because that it is the recipient's body which has this immune system and the immune setup with these all these antibodies in them. Okay, so when we talk about the mother here as well as the system, that is the host system, it has not seen the R positive protein. And we must also recognize the fact that this is not a two-way communication during pregnancy. So this is in fact a one-way communication where the fluids and the nutrients are transferred from the mother to the child. Okay, it's not the other way around. So basically, their blood does not mix until childbirth. Okay, so it is during childbirth that their blood may come into contact and mix up. But before that, prior to that, their blood does not mix up. So your mother does not see this protein until the childbirth. So until the childbirth, this is not a problem. There are no antibodies generated by the mother's immune system because it has not seen this R positive. So it is during childbirth that there is some blood mixing and blood ends up mixing with each other. And that's when this protein is exposed to this immune system of the mother, and then the mother generates that response. So during the childbirth, the mother develops these anti-R positive antibodies. But as far as the childbirth of this or the health or progress of this child is concerned, it gets unaffected. It is not affected. The Rhesus disease is about the subsequent pregnancies, the subsequent births, the births of the second child, for example, or the third child. That is where the problem is. Because during the childbirth, during the mixing of blood, for some reason, the mother has now developed these antibodies. Okay. So now the mother's system has these antibodies, and in the second pregnancy or in the second birth of the second child, that is when this is going to become an issue, because now the mother has already developed these antibodies and it recognizes these Rh proteins as foreign objects. So now when the blood comes into contact, it will definitely attack it. And because those antibodies have been soundly or proficiently developed until then. Okay, so this is a problem. The Rhesus disease is a problem for second, third, or subsequent childbirths, and that is where this is a huge issue. And if and the resolution for this and the solution to this is that there are certain chemicals, certain medication that could be taken in order to in order to stop the production or stop the response of the immune system when or during this the period of uh, subsequent pregnancies, so that the mother's immune system does not affect or respond to positive protein. So you could take certain chemicals, certain medication that would suppress that immune response. So that is what we what we figured out. As a quick recap, I'm going to summarize this again. Look, the rhesus disease is about mother being R negative, father being R positive, them conceiving a child having R positive rhesus protein. This we know from the basic fact that R positive is a dominant allele and it expresses itself as a phenotype. So the child will definitely have R positive protein. Mother being R negative is going to conceive it, is going to see it as her immune response is going to see it as a foreign object, and it should affect its progress, health, and should basically destroy those cells. But this blood does not mix until childbirth, and that's the first time when the mother's immune system sees these as a foreign object and develops those antibodies. During the first childbirth, it is not a problem, but in subsequent childbirths, because in the first childbirth, it has developed those antibodies. The second or the third, for the second or the third childbirth, that is when it's going to become a problem, because now the mother has developed antibodies. Now, when the mother's cells are nutrients and um, the fluids are going to be fed in to the child, that's when it's going to be a problem, because the mother has now developed those antibodies. It has antibodies against this protein. So the second pregnancy is going to be a major issue. It's going to be a big issue. Okay. So to avoid that, what basically is done is you end up taking certain medications or chemicals to suppress that immune response, to make sure that these proteins are not affected by mother's antibodies. Okay. So that is what was the rhesus disease, and that is what we discussed today. Towards the last few minutes, we talked about certain examples for students who had an issue with the Lab 9. And if any of you want to discuss those, happy to discuss. If there are any questions related to this lecture, happy to discuss them. Please let me know. Are there any questions?
Sir,
Sir,
second
Go ahead, one by one. Go ahead.
सर सेकंड बर्थ चाइल्ड के अंदर जब हमारे पास एंटीबॉडीज को वो प्रोड्यूस करना शुरू करेगा मदर, तो उसमें तो ऑटोमेटिकली फिर ये होगा कि बेबी के अंदर सारे रेड ब्लड सेल्स डिस्ट्रॉय होना शुरू हो जाएंगे.
एब्सोल्युटली एब्सोल्युटली, यही तो मसला है कि सेकंड चाइल्ड बर्थ की इवन ड्यूरिंग प्रेगनेंसी, क्योंकि रिमेंबर जो फ्लो है, फ्लो ऑफ न्यूट्रिएंट्स या फ्लो ऑफ फ्लूइड्स या जो भी डेवलपमेंट ऑफ द चाइल्ड, ऑफ द बेबी, दैट इज थ्रू मदर. ओके, सो after the first pregnancy, the mother has developed those antibodies. So she has those antibodies, and those are going to, somehow or the other affect the child during its development in pregnancy and during childbirth. So it is absolutely fatal. That's when the blood gets mixed. So second pregnancy mein, it's absolutely fatal. So what they do is, before the childbirth, during second pregnancy, they treat the mother with medications and everything to ensure that the immune response is suppressed. Immune response does not come into play.
Okay, sir.
Right. Any other question? Well, I think we had a question. I think Jazati. I don't know. Okay, if there are no other questions,
my question was same as Fajr, so I'm clear.
Okay, ji, that's good. I think we are going to conclude our session now. If you had any issues with the Lab 9, which was about finding the projection, because that is important. The projection part is, that is part of the final exam. So if there are any issues regarding that, you are welcome to ask. Because the majority of the class did not do well on that. So we had a brief discussion on this. Basically, I was telling students to take the rectangle's bottom right corner as a point of reference for computing your angles. It helps to make the triangle simple for small angles like 5, 10, 15, 20, 35, 45. And take the top right corner for angles above 90. That makes the triangle computation simpler. if you take this point for the 95 angle from the
ये
horizontal,
रिपीट आज से बता दें कि 5:15 के लिए हम लोग कहाँ से करेंगे और दूसरे
this is the original rectangle. Bottom right corner rectangle का ये है. If you do a 5-degree line from here and then draw a perpendicular line for sampling, तो ये 5 degree है, this is 85 and then this is again 5. तो once you get this 5, तो ये triangle आपको मिल जाता है. Okay, तो this आप bottom right corner जो rectangle का है वहाँ पर draw करें अपना point of reference for small angles, तब ये triangle बड़ी आसानी से मिल जाती है आपको. But for bigger angles like 95, 100, 110, उसके लिए आप top right corner choose कर लें. Once you choose the top right corner, तो ये horizontal से 95 या ये 100, 110, 20 के angles बड़े आसानी से मिल जाते हैं, वही आसानी से बन जाते हैं. एंगल ऑन स्ट्रेट लाइन वो 180 है, तो आपके पास रिमेनिंग एंगल यहाँ आ जाता है, और रिमेनिंग एंगल जब यहाँ आ जाता है तो ये परपेंडिकुलर है, तो 90 में से आप उसको माइनस करके ये अंदर वाला एंगल मिल जाएगा. once you get this inner angle once you get this inner angle, this triangle is fairly simple to make and find the unknown dimension.
Excuse me, sir.
जी
सर रेडॉन ट्रांसफॉर्म एक्स-रेज में जो एक टॉपिक है उसमें से आप पेपर में कैसे दे सकते हैं? मतलब आई एम नॉट गेटिंग हाउ टू प्रिपेयर इट. सर उसमें सारी वो इंटीग्रेशन वाली इक्वेशंस हैं, उनको रट्टा लगाना है या उनको कैसे प्रिपेयर करना है? सर एक्चुअली मैंने आपको वो लेक्चर मिस कर दिया था और क्विज भी मिस हो गया था, और मेरी उस टॉपिक की इतनी अच्छी प्रिपरेशन नहीं है. आई एम वेरी वरिड अबाउट इट. मतलब एक्सरे मैंने प्रिपेयर कर लिया है सारा, लेकिन उसमें रेडॉन ट्रांसफॉर्म समझ नहीं आ रहा.
what I remember X-ray में आप, and I think there are notes on, maybe there are no notes, but उसके अंदर we had a, उसकी वीडियो भी है on the on YouTube and on our course files page as well. उसमें एक्सरे के अंदर we said कि we don't do the integration, ये हम इंटीग्रेशन ही कर रहे हैं, जो हम यहाँ से इसका डिस्टेंस फाइंड कर लेते हैं, बेसिकली ये इंटीग्रेट ही हो रहा है. So this is what is expected of you to make these projections. आप projections बना सकें, उनकी projection की जो shape है वो बता सकें, उसका amplitude maximum बता सकें. You should be able to know that. The lab 9
सर
basically.
ये तो आता है, प्रोजेक्शंस का मतलब कर लिया था, एक फ्रेंड ने करवा दिया था, बट जो रेट ऑन ट्रांसफॉर्म की इक्वेशंस हैं वो किस तरह से, मतलब उनको प्रिपेयर करें या नहीं करें, उनको कैसे करना है?
You're not required to memorize or rote learn them. आपको बिल्कुल रट्टा लगाने की जरूरत नहीं है. This is what we do. उस equation को solve करने की बजाय यही तो वो काम हो रहा है यहाँ पे. So I will not ask you to write down those equations or solve them. ये हम उन्हीं पे
ओके
solve करें यहाँ पे, यही तो हो रहा है solve. The other thing
ओके
yesterday was the intensity calculation, कि कितनी वो उसमें से attenuation हो जाती है when it moves from
यस.
source. And other than that, please just go through the video. It will be very much similar to what we did in class in the video. I could.
सर कोर्स फाइल्स पर है वीडियो?
जी वहाँ सारी वीडियोस हैं, आप
ओके सर,
क्लिक
थैंक
करेंगे
यू.
तो link है. Okay, I think there are no more questions, so we will stop our discussion today, and we'll see you on the final exam. Thank you so much, Ji. If there are any questions, you're welcome to stay. Otherwise, you're welcome to go. Thank you so much, Ji.